Thank you very much. It's a pleasure to be here. And I'm representing Novai, which was founded in February 2020. Like a lot of the companies here, one of the things that we are trying to deal with are those aging diseases that are leading to vision loss, specifically macular degeneration and glaucoma. Now, the problem tends to be that in these diseases, we identify patients a little bit too late. So they've already lost quite a bit of their vision by the time you start treatment. So I'm thinking here, for example, of functional changes, the visual field and glaucoma, OCTs before that. But ideally, you want to try and get those patients before any vision loss has occurred. So this is our solution. It's the dark molecule, which is an annexin, which is has a fluorescent tag on it. And it identifies cell membranes that have been disorganized because the cell is either stressed or going into apoptosis. So using the transparent nature of the eye, what we can pick up are the spots that become fluorescent when, after giving an intravenous injection, it enters into the eye. And you can see in this picture here that a patient's retina has these white spots, which clearly can be delineated by the blue circles. The blue circles are actually our AI algorithm, which is automatically identifying those white spots. Now, in addition to detecting those white spots, the other reason why the AI is incredibly important is it gives a robust idea of what those white spots are. It can allow you to segment those, so you could look at the morphology of those spots, and that all can help you decide whether or not, firstly, your patient is responding to treatment, or whether they are going to go on to progress. And that's important because you know which patient needs to be targeted and whether they are on appropriate treatment. And ultimately, and this is what we're doing now, we're working with Onl Therapeutics, for example, is we're also assessing very rapidly patients' response to new treatments. So this is an example of some patients that we've had with geographic atrophy, with glaucoma, and with optic neuritis. And you can see here the red squares are identifying the AI cells that have been labeled with our dark molecule. I think one of the things that is incredibly important with new drugs coming in is the ability to predict future progression. And over here, what you can see is 35 months apart, the degree of atrophy at a baseline level in a patient, and then the final GA atrophy. And if you look at the overlay, the bottom right square, what you will see is, in addition to predicting future growth, what dark is doing here is also predicting the direction of growth. And so if you've got an extrafoveal lesion, this could be very valuable indeed, because it would identify which patients are at risk of losing vision. This is showing you our published paper from the Glaucoma Cohort in our phase 2A. And there are two things I want to point out here. So dark was only performed once. Patients were divided by OCT results, RNFL thinning, at 18 months later as to whether they were stable or whether they had progressed. So there was a significant difference in the number of dark spots, but also, and this is highly unusual in a biomarker, if you provided a cutoff, and here we've put a cutoff of 30, all the patients who had a dark count greater than 30 went on to progress 18 months later. So that's an enriched population, but also identifying those at risk of future vision loss. We've been able to develop a number of commercial partnerships. As I said, we started just before COVID, so we had to learn how to do this working with pharma partners remotely. And we are furthest ahead with our programs for macular degeneration, GA included, and glaucoma. We've also very recently started diabetic retinopathy studies and due to be starting inherited eye disease. Our philosophy that the eye is a window into the brain is also being shown by various publications, including preclinical and clinical. And we're using grant funding to look at patients with optic neuritis and MS, Parkinson's disease, we've just had a grant for Michael J. Fox, for example, and Alzheimer's disease. So our IP portfolio is getting stronger all the time. We have some patents originally licensed from UCL, but we have our own, as in Novai's, and this is an area that is expanding as well. The market we are going for initially is this serviceable, obtainable market, which we reckon is around 6.6 billion, and that's a conservative estimate. But as you move along in the circles with the addressable markets, you can see that the amount that we're talking about in the market is quite sizable. So currently we are at this first stage. We are being used as an exploratory endpoint, and we've raised around 4.2 million in terms of sales contracts, working with pharma partners to try and accelerate their drug development. Where we want to be, and this is what we're looking for money for now, is recognized as a surrogate endpoint so that we can be integrated as a clinical decision support tool. And that will be allowing patients and clinicians to basically be put on the correct treatment pathway, the most appropriate treatment for that person, but also to avoid them ever losing vision before that treatment is commenced. And ultimately, we see ourselves being used as a screening tool. So currently this is given intravenously, but we are going into a nasal formulation. This is our leadership team, and Alex Wilson is here with me. And we've been very fortunate with a large group, and all of these people I think you will recognize as our KOLs and advisors. We're moving on now to start the phase 2B to allow us to get this registered as a surrogate endpoint. And we'll actually tranche this into the various phases of our development. First, getting into a surrogate endpoint approval, then becoming software as a medical device as a clinical product development, and then finally going to market. So I'm really hoping that we can have some good discussions as we are able to talk to people in the networking sessions. And once again, many thanks to OIS for inviting us. Thank you.